Hello दोस्तों, welcome to another tutorial. Today we will discuss about the DNA replication. As we know that the DNA replication is the process of formation of DNA from a DNA, or we can say that the exact replica of DNA is produced from DNA template. So the formation of copy of a DNA, that is known as DNA replication. In previous videos, that is replication 1 and the basics of DNA replication, we will discuss about the enzymes required for the DNA replication. Now we will go with the process. We will discuss about the process. As we know, the DNA replication can be divided into three steps. How many steps? Three. And the three steps is, one is the initiation, first, second is the elongation, and third is the termination. So these three steps are occurs during a whole DNA replication process. Let us discuss one by one. The first we will discuss about the initiation of DNA replication. It is very typical process. It is not just easy as it's look like. First of all, we will already discuss, but again I will revise. There is a requirement of proteins for DNA replication: DNA A, SSB, DNA B, DNA C, DNA G, DNA polymerase enzymes, etc. Let us discuss. Let's have a DNA. As we know that we will discuss about the how many origin of replication sites are present in prokaryote and eukaryote in previous video. Let us take about first about the prokaryotic DNA replication. As we know, the DNA of prokaryotic cell is circular, and we take it is in produce it linear, and we saw that there is a origin of replication site which is 245 base pair long, and it contains 3 units of 30mer, as we discussed in already in previous video, and 5 units of 9mer in this 245 base pair DNA, and a DNA A protein come and bind to these 13mer units, come and bind to this 13mer units of a DNA stand in an origin of replication site, ORI site. It also interact with the 9mers. It bind to 13mer and also interact with the 9mer units. Okay, and it it required divalent ions, Mg plus ions or Zn plus ions, and interact with the hydrogen bond in the 9mer units, hydrogen bond, and also with the ATP. It break down the ATP molecule into ADP, into ADP, ADP, and it cannot bind to ADP. So it only bind to ATP, and it will break down it into ADP, and the energy is released. And this energy is utilized in break down the hydrogen bond between two DNA strands, and due to which a bulge is open like this. A bulge is produced, which is known as replication bubble. It is known as replication bubble. The hydrogen bond is breakdown. And as the DNA protein come, as I already told in last videos, when the DNA protein come, it bring itself with another proteins, DNA B and DNA C. DNA B and DNA C. DNA C is known as loader of helicase, loader of helicase, helicase. And DNA B is itself known as helicase. So it load the helicase in the melted DNA part like this. And the helicase also have a six subunits. Helicase also have a six subunit, and it come and bind to this DNA strand. And the hydrogen bond is breakdown. That is known as the replication bubble. As the DNA helicase is loaded between the DNA C is generated from this strand, and another protein, which is known as the SSP protein, come and bind to this single stranded DNA. This single stranded DNA. So the DNA could not join together. The hydrogen bond is not produced. It stopped the hydrogen bond formation between the two melting DNA strands. Now that this DNA is remain like a open like this. And now another protein which we discuss, that is known as the DNAG. We will discuss in previous, which is a RNA primase enzyme or polymerase. That is known as the RNA polymerase. Primase or RNA polymerase enzyme come and bind to this helicase units. I'll use different color for this. It come and bind to helicase on this melted DNA strand. And this structure from where this DNA is melted, that is known as replicon. Replicon. It is known as replicon. Now, first, when it is only melted, then it is known as the replication bubble. And where the replication is occurred, that site of replication is known as a replicon, like this. Okay. So, abhi, that is the RNA primase is loaded here. Now, what happened next? What happened next? Let's see. This is the open DNA strand, like this, like this, and here is the helicase. Here is the helicase unit, and it combined with the primases like this, and SSB is present there. This complex of helicase and this primase is known as primosome. What is there? Helicase plus primase. RNA primase. It's commonly known as primosome. Now, this primase enzyme have very less affinity and it come and bind for a little bit time and it gets dissociated from here and leave a 15 to 35 nucleotides. It's left the DNA after after leaving after leaving the A stand of RNA on a DNA template like this. If we see this, it is the 5 prime, it is the 3 prime. Here and here like this. We see this. So, as the DNA is on this template, is moving on this direction, if we see it in rubbing this like open completely, so what we saw 3 and 5. The direction of this RNA formation as the DNA direction of DNA replication is always 5 to 3 and using the 3 to 5 template. So direction of this RNA formation is like this towards this and or of this is like this. Okay. When this RNA is produced over here, now the another protein which is known as the DNA polymerase. As I already told about the polymerase in last video. So the DNA polymerase third in prokaryote come and bind to this site with the help of its with the help of its beta clamp over this DNA strand and its polymerization site is at 3 prime. Similarly here and this is beta clamp is here and it is joined with the help of a toy unit like this. है ना? And similarly here another RNA polymerase is joined and it have also a toy unit and here is also a toy unit and its beta clamp like this. A structure is produced here like this. Okay. So two subunits of DNA polymerase come together and join the DNA strand like this. Okay, now clear? However, in some standard book we found that key leading strand is produced 2000, 1000 to 2000 base pair. Then after the lagging strand formation is started and let's discuss about these thing also later on. First, we know that the assembly of DNA polymerase on a DNA template is like this. Now, what happened? This polymerase move in this direction, and this polymerase move in this direction. Similarly, this polymerase move in this direction, and this polymerase move in this direction. So, after moving some apart, after moving some apart, what happened? What happened? Let's draw another diagram here. Oh, what happened here? What happened here? Helicase reached there. अभी हम सिंपली बना लेते हैं. Helicase reached there, and the RNA primer, which is here, is here and here, and another RNA primer is here and here. Its direction is 5 to 3. Its direction is 5 to 3. Its direction is 5 to 3, and its direction is the 5 to 3. ओके, ये भी खोल ही देते हैं, नहीं तो समझ में नहीं आएगा. ढंग से कर देते हैं इसको. सिंपल करते हैं इसको. And this is the helicase, and this is the helicase, है ना? And this is the 5, this is 3, this is 5, this is 3, and DNA template is producing after this. So this DNA polymerase moving by forming the DNA on this side, and it reached to here. Similarly, this DNA polymerase moving in this direction, and it reached to there, here. First of all, let's see it. When the toe unit is stretched, so this toe unit is breakdown. जब ये दोनों एक दूसरे से पार्ट इधर की तो A1 moving in this direction, other moving in this direction, so there is a stretch in the toe unit and their conjugation is breakdown. And another option is that there is this polymerase found the RNA in front of it, and it's leave the that side. So that RNA polymerase leave it, the DNA. That RNA polymerase leave the DNA after reaching near the RNA. Okay, here. Clear? And this will not leave, this will move forward. This DNA is moving forward in this direction because it do not found any obstacle. Helicase is moving in forward direction, and it is follow the helicase. Similarly, this DNA is also going to this direction and found RNA polymerase, and it also dissociated, and this DNA is also moving in this direction with polymerase, and it does not found any obstacle, so this DNA strand is moving forward. That the situation, this condition is known as replication fork. This condition is known as replication fork. When the DNA is DNA polymerase is assembled, RNA primer is present there, and the DNA start forming the DNA stand. DNA polymerase start forming the DNA stand. That is known as the replication fork, and the end of the initiation. We will saw this in this diagram. This is the polymerase enzyme, and we will saw it like this. These are the helicase binded to the this site like this. It then the SSB protein come and bind to this site, and then DNA RNA primase come and join with this, that is known as the primosome complex, and a complex is produced. After the DNA is produced, so the arrangement is like this. These are the two tau unit which is joined together and forming polymerase. Catalytic properties are there and there. So one DNA strand is moving in this direction and another is moving in this direction. And there is the helicase moving apart. So this DNA strand is produced continuously. And this DNA strand produce in small fragment like this. पहले ये बन गया, फिर अब ये बन रहा है. तो पहले ये बन गया। तो ऐसे छोटे छोटे सेगमेंट्स में डीएनए का स्टैंड बनता है। स्मॉल स्मॉल सेगमेंट्स आर प्रेजेंट एंड दिस कंडीशन of formation of leading and lagging strand is discovered by Okazaki. Okazaki, a Japanese scientist, and he told that these small fragments as an Okazaki fragment. Okazaki fragment which contains the DNA strand of 100 to 200 nucleotide base pairs. तो उन्होंने ही बताया, he was the scientist who told that the DNA replication is not occurs continuously. It occurs in small fragment on a one strand, and one strand is produced and on the basis of regular. So like this, this condition is occur. Now what happened in elongation? At that state, the helicase again requit another primase here and another primase is here. So there is the tendency or requirement of primase for every lagging stand. So now this again a primosome complex is produced. Now it will produce again the RNA primer here, and the again the RNA primer is RNA primase is goes leave this site and goes away. RNA primase go leave this site with RNA and again goes away, and again the DNA polymerase join this site, and this site again the DNA replication occur in this direction, and again this DNA replication occur in this direction, but it does not affect this stand. This stand is continuous and this stand is continuous. So that is known as the elongation phase. That is known as the elongation. What happened next? What happened next? Like this. This is the DNA stand. Complete. This is the complete DNA stand. And this is another. And now what we saw there, this is the site from where the DNA replication is started. This is the 5, this is the 3, this is the 5. Sorry, this is 3 and this is the 5. And its 5 to 3 is this. And its 5 to 3 is this, and its 5 to 3 is this. What we saw, the DNA replication occur on this stand is continuous like this. And what we saw, this DNA replication occur on this site is continuous like this. Okay. Now what happened on another site like this? The meantime there is RNA primer is come and produce, and this DNA is produced first this, then second this, then third this, then fourth this. Similarly, conditions occur here. First there RNA, then again RNA primer, then again RNA, then again RNA, and its direction is like this, 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 and this. These are the direction. So one stand produced completely, that is known as the leading stand. That is another leading stand on this direction. And these small fragment which contain the RNA and DNA hybrids is known as the lagging stands. It is known as the lagging stand. That is also known as the Okazaki fragment. Okazaki fragment. Now, as I already told, the DNA of a prokaryotic cell or Escherichia coli have circular. So the newly synthesized DNA is present on there. That is like this RNA primase, and this is the DNA, and one strand is complete, a half. Similarly, here The DNA is completely where the small fragment are present, but the another fragment from where the DNA is produced in very small amount, like this. ऐसे बनेगा बीच में दोनों के एक मतलब एज वी नो द डीएनए रेप्लिकेशन इज सेमी कंजर्वेटिव, वन स्टैंड इज पेरेंटल एंड वन स्टैंड इज डॉटर, तो ये पेरेंट का हो गया, ये डॉटर का हो गया, ये पेरेंट का हो गया, ये डॉटर का हो गया, है ना। तो ऐसे बनेगा। अब क्या होगा कि अब दैट इज टर्मिनेशन। क्या होगा टर्मिनेशन में, जब डीएनए इस तरीके से पूरा बन जाएगा, नाउ व्हाट हैपेंड, द डीएनए पॉलीमरेज 1 कम, नाउ द डीएनए polymerase 1 come, जैसे I already told कि there is 5 type of DNA polymerase are present, तो DNA 1 says that the you go and produce the DNA, I will handle the any problem. That is the words of DNA polymerase 1 to the DNA polymerase 3rd. Now this DNA polymerase 1 come and remove these RNA fragments from here. Replace, it's replace. कैसे करेगा replace? Like this, कि जैसे ये RNA था, तो ये इस आरएनए को हटा देगा यहाँ से ऐसे, और इसकी जगह पर यहाँ पर बना सॉरी इसकी जगह पर यहाँ पर बना देगा ये डीएनए। लेकिन ये जो लास्ट वाला न्यूक्लियोटाइड होता है, जो कि एक न्यूक्लियोटाइड तो है डीएनए का और एक न्यूक्लियोटाइड है आरएनए का, ये एक हाइब्रिड है डीएनए और आरएनए का, इसको नहीं हटा पाता ये डीएनए पॉलीमरेज 1। सो इट रिक्वायर्ड फैन न्यूक्लिएजेस, fan 1 nucleases, it the enzyme which have capability to break down the DNA RNA hybrid. So this come and break down this DNA, and then again the ligase enzyme come and join these DNA strands, and the RNA primase is degraded by RNA enzyme. Now similarly this RNA removed, and the whole DNA is continuously synthesized, and these fragments are joined together by DNA ligase. DNA ligase enzyme, which for ligate by the making the phosphodiester bond between uh, forward and backward nucleotides. That is the termination. So this is the whole diagram which show the DNA replication in a single diagram. What we saw there, this DNA strand is produced continuously, that is known as the leading, and this DNA segment is produced like this in small fragments like this. So this is known as the Okazaki fragment. So DNA replication is occur like this. Now there is the last option. There is another enzyme which is we not discuss in previous video, and this is, so we discuss it here, is that is the topoisomerase. What will topoisomerase do? As we know, the DNA strands are open. The coiling of DNA strands are opened by helicase. So it produced a positive supercoiling. Positive supercoiling. तो जब पॉजिटिव सुपर कोइलिंग इज प्रोड्यूस हेयर, तो द डीएनए इज व्हेन वी ओपन टू डीएनए स्टैंड्स लाइक दिस, तो एक पॉजिटिव जो दैट पॉजिटिव सुपर कोइलिंग में कॉज नोट फॉर्मेशन, गांठें बना देगी नोट, and once the note is come in front of the helicase, it will not move further, ये आगे नहीं बढ़ पाएगा as the note come in front of the helicase, and it will dissociate from the DNA strand, and DNA replication should be halted, रुक जाएगी। तो ऐसा ना हो, उससे बचने के लिए क्या होता है, एक टोपोमरेज, टोपो आइसोमर, टोपो मीने टोपोलॉजी, आइसोमरेज मीने उसको एक जैसा, समान, आइसो, आइसो मीने सेम करना। तो दिस टोपो आइसोमरेज क्या करेगा, इस डीएनए के स्टैंड को, मतलब देर इज टू एंजाइम्स फॉर टोपो आइसोमरेज, टोपो आइसोमरेज 1, वैसे तो बहुत सारी कैटेगरी, एंड 2, तो दैट इज 1 क्या करेगा, इस डीएनए के एक स्टैंड को तोड़ देगा, इट ब्रेक द डीएनए 1 strand and it need ATP molecules, or it free the DNA molecule from this template and produce a negative supercoiling. It produced a negative supercoiling. So note is breakdown, the DNA polymerases move further. That another enzyme is topoisomerase 2, which break down the both DNA strand and produce negative supercoiling again. By breaking the two, it required NADP molecules. So topoisomerase break down and again join these DNA molecules. Again join these DNA molecules after producing the negative supercoiling. So topoisomerase have two properties. It have an helicase and it have an ligase, or sorry, or restriction endonuclease property. It have these three properties. It work as an helicase, it work as an restriction endonuclease, and it work as an ligase. So that is all about the DNA replication. Like this, the DNA primers are removed here like this, and new DNA strand is produced. The fan one cleavage the primer, and that is all. Huh. Now we will discuss about the difference between DNA replication of prokaryote and eukaryote. There is not much difference, तो we will discuss here. देखो वहां पर हेलिकेज एंजाइम इज, हेलिकेज का काम कौन करता है, इकोलाई में डीएनए बी, यहाँ पर एक प्रोटीन होती है जिसको बोलते हैं एमसीएम, मिनी क्रोमोसोम मेंटेनेंस कॉम्प्लेक्स बोलते हैं इसको, मिनी क्रोमोसोम मेंटेनेंस, एंड फेजेस में इसको बोलते हैं 41। ठीक है, तो एमसीएम का मतलब क्या है कि मैं जैसे मैं ऑलरेडी वी विल डिस्कस इन द प्रीवियस वीडियोस, दैट इज द इन यूकैरियोट डीएनए रेप्लिकेशन अकर एट मल्टीपल साइट्स। तो मिनी मिनी छोटी छोटी जगह पर, बहुत सारी जगह पर डीएनए रेप्लिकेशन चल रही होती है, तो दैट इज द एमसीएम कॉम्प्लेक्स की जरूरत होती है। लोडिंग यहाँ पर डीएनए सी करती है, तो यहाँ पर लोडिंग कराने के लिए कौन आती है एमसीएम को, CDC6। यहाँ पर जो डीएनए के स्ट्रैंड खुल जाते हैं उनको बाइंड कराने कौन आती है, एसएसबी प्रोटीन, लेकिन यहाँ पर कौन आएगी, आरपीए, और फेजेस में 32, और यहाँ पर 59 CDC का काम करेगी। प्राइमरी के लिए यहाँ पर कौन आती है, डीएनए जी, तो यहाँ पर आता है polymerase alpha. I already told about the polymerase alpha in previous video. That is the only enzyme which can produce RNA as well as DNA when it come to the DNA template. तो ये 15 से 35 न्यूक्लियोटाइड आरएनए बनाता है, फिर उसके बाद डीएनए रेप्लिकेशन को शुरू कर देता है। यहाँ पर स्लाइडिंग क्लैंप का नाम था बीटा क्लैंप, बीटा। ठीक है, यहाँ पर है पीसीएन, क्या नाम है यहाँ पर, पीसीएन। पीसीएन का मतलब क्या है, प्रोलिफरेटिंग सेल न्यूक्लियर एंटीजन, तो इनकी जरूरत होती है। ठीक है, और यहाँ पर इनको क्या बोलते हैं, 45 फैक्टर। क्लैंप लोडिंग का काम कौन करता है, एटीपी इसको खा करके, गामा क्लैंप लोडर, यहाँ पर इसको बोलता है आरएफसी, रेप्लिकेशन फैक्टर सी। कैटालिसिस पॉलीमरेज थर्ड के द्वारा होती है। यहाँ पर मैंने आई ऑलरेडी टोल्ड दैट द पॉलीमरेज डेल्टा एंड पॉलीमरेज एप्सिलॉन आर देयर रिक्वायर्ड। तो पॉलीमरेज एप्सिलॉन जो होता है, ज्यादातर स्टडीज में देखा गया है कि इट विल प्रोड्यूस द लीडिंग स्टैंड, लीडिंग स्टैंड। ठीक है, एंड द पॉलीमरेज डेल्टा प्रोड्यूस द लैगिंग स्टैंड। तो in eukaryote there is two different enzymes are involved to produce leading and lagging strand, ये बात याद रखें। रिमूवल ऑफ आरएनए यहाँ पर पॉलीमरेज 1 करता है, यहाँ पर फैन 1 करता है, और यहाँ पर 43 करता है। लाइगेशन का काम यहाँ पर लाइगेज एंजाइम करता है, यहाँ पर 1 करता है, यहाँ पर T2 T4 ligase करता है। तो ये छोटा सा डिफरेंस है, बाकी डीएनए रेप्लिकेशन इज सिमिलर इन बोथ यूकैरियोट एंड प्रोकैरियोट्स। मैनर जो होगा वो सिमिलर तरीके का होगा, बस ये छोटा सा डिफरेंस है। यहाँ पर जो प्रोटीन्स हैं, जो एंजाइम्स हैं, उनके नाम अलग हैं, दे हैव डिफरेंट नेम्स। So this is all about the DNA replication. I hope you will understand it and you will carry on to watch our channel. Thank you so much. If you come to watch here, kindly subscribe it, and it, we will start the new series from 1 March about the NCRT of class 12th and class 11th for the NEET, and it also will be useful for JRF students, CSIR JRF, and for the PGT biology, RBSE, HBSE boards. So be in touch. Thank you so much.